Hello, everyone. My name is Kevin Schug, and I'm a professor at the University of Texas at Arlington. I'm very excited to be here today to tell you a little bit about the research we've been doing on hemp, low THC cannabis, specifically in this case using liquid chromatography and tandem mass spectrometry to look at anthocyanin flavonoids, those molecules that give the plants the various different colors, and how those vary between different cannabis cultivars. I want to first, recognize Mike Pecor, as he is one of my students who's done the majority of this work. This is a picture of my group from the not too distant past, and also the collaboration with Z Therapeutics for providing the hemp and conducting some of the time course study that I'll talk about at the end. So let's get going. I'm just going to very quickly review some typical general cannabis analysis, right? Just to remind you of the analytical techniques that are used commonly, and then we'll go into talking specifically about LCMS and its application to looking at these anthocyanin flavonoids, and ultimately looking at those inside the different hemp cultivars. So I think anybody who's been, in and around the cannabis industry knows that there's a very standard set of methods that are commonly needed to be applied for quality control of cannabis, and this just starts simply with, being able to measure moisture. Obviously, the main bioactive compounds in these plants are the cannabinoids that we're interested in, and the various different plants have various different levels, and we have low THC hemp and high THC cannabis, and so we need to be able to analyze the potency of these plants. That's typically done using HPLC, so liquid chromatography, the UV detector. It's also common to want to look at terpenes. These are also bioactive compounds that convey the flavors and fragrances of the cannabis plant, and that's mainly done with gas chromatography with the FID detection. Pesticides gets a little bit more complicated. We need a little bit more power with our analysis in order to really see trace levels of these pesticides. And different pesticides may be more or less amenable to using gas chromatography or liquid chromatography. But again, typically needing mass spectrometer there for more specificity and sensitivity in the detection. And depending on your state, obviously we should hope that people are testing these things regularly and across the nation, but use of things like ICP mass spec for metals analysis, and a variety of typical biochemical methods would be used to look at the contamination of plants by microorganisms like mold and bacteria. Okay, so that's kind of the landscape that you all are interested in and used to using. I'm going to go a little bit more depth into a technique called liquid chromatography tandem mass spectrometry. This is a technique that uses a fancier mass spectrometer, a triple quadrupole mass spectrometer. The combination of these techniques really is already the gold standard for trace quantitative analysis in a lot of areas, including clinical analysis, looking, as we've already mentioned, pesticides, but labs use these things very regularly to interrogate biological species, peptides, lipids in very complex mixtures. This is a really highly precise and efficient separation technique for non-volatile and semi-volatile compounds, coupled to a detection technique that gives you very good sensitivity and very good specificity, and I'll elaborate on that a little bit more. The front end of the instrumentation is your HPLC. And really, this is not much different than what you might see in these more simplified, let's say, cannabis analyzer here. This is a product that Shimadzu provides, where we have the ability to inject very small volume extracts. We have a system where we have high pressure pumps delivering a mobile phase that allows you to separate the compounds as they pass across a stationary phase. In this case, usually something like a C18 functionalized particle packed in this 10 centimeter long column. And as I said, typically UV vis detection is good enough for potency analysis, but when we really want to go and look at things more in depth and at lower levels, we need to be considering a different detector. And so the triple quadrupole detector here features an electrospray ionization interface. This is the way that we convert molecules in the liquid phase into gas phase ions. They need to be in the gas phase for us to manipulate them in the mass spectrometer. And then I mentioned tandem mass spectrometry. This just refers to our ability to take those ions once they're in the gas phase and fragment them further to learn more about them. It's kind of like, you know, taking apart a jigsaw puzzle. These things will break at different points, and we can use those fragmentation as a kind of diagnostic tool to characterize the molecules. So overall, you know, we can operate this instrument in many different ways, and we're going to utilize several of these techniques as we go forward. So looking up, you know, if you were to look under the hood of this triple quadrupole mass spec, what you see is what you might expect, you know, 3 quadrupoles. And they are arranged here in this schematic where we have the electrospray ionization source. You have a single first quadrupole that allows you to select different ions or scan across a range of ions. The second quadrupole is used exclusively to induce collisions to fragment the ions of interest. And then we can use the third quadrupole to sort those fragments. So for example, a really common way to use this technique would be to use what's called a product ion scan. So if you see there on the left, we have this mixture of M+ ions that are fed into the first quadrupole. We're interested in just one of these, this M2+. So we tell it to select that ion. Maybe it has a massive charge of 450. It goes in the second quadrupole and is fragmented into different fragments. These are these puzzle pieces. And then we can use that third quadrupole to sort of sort those puzzle pieces and generate a mass spectrum that would be diagnostic of that initial product of interest. So this gives us good qualitative information about an ion of interest. This is you know, the same type of approach, at least with the first quadrupole, is utilized when we go into a multiple reaction monitoring mode. And this is really where the power of triple quad mass spectrometry comes for quantitative analysis. This multiple reaction monitoring mode allows really great sensitivity and specificity. So just like the last one, we have this mixture of ions. We use the first quadrupole to select an ion of interest. This is called the precursor ion. So that's the molecule, you know, we may be interested in quantifying. And when we develop the method, what we'll do is we'll fragment that through Q2, generate a set of fragments, and then pick a particular fragment for the third quadrupole to monitor specifically. When we do this, we are operating in the most sensitive manner for these quadrupoles. So when we have it sit and look at a specific ion at one time, this is the most sensitive way that we can utilize this instrument. When we scan across multiple ions, we do lose some sensitivity. Now, in this case, because we're measuring a specific product of a specific precursor ion that we've chosen, we gain lots of specificity because there's not likely many other things in that mixture that would generate that diagnostic product ion. Additionally, the extra sensitivity comes because we really reduce a lot of noise from the analysis with this kind of triple filtering mechanism that we have in a triple quadrupole. So that effectively, that lowering of the noise allows us to have a higher signal to noise ratio, which is, you know, what we're looking for quantitative analysis. One other approach that's used with these instruments that's really less utilized is what's called a precursor ion scan mode. And this is a bit different. This is used to discover molecules in the mixture that have common fragments. So in the previous two, we were just told quadrupole 1 that we're only interested in one ion. But in this one, we're just going to have quadrupole 1 to sort our ions in order of increasing mass to charge ratio. And then each one of those, as it comes through the first quadrupole, is subjected to fragmentation and creates its own series of fragment ions. But we tell quadrupole 3, we're only interested in seeing molecules that generate a particular fragment. In this case, this F2+, you know, something that has a mass to charge of 335. So we will only register a signal at the detector when something comes through and is fragmented and generates this 335 ion. So we choose this ion to be something diagnostic of, let's say, a class of compounds that we're interested in seeing. And then by running in this mode, we can now go and see if there's something in the sample that generates this fragment ion. When it does, that fragment ion is timed with the same, we can go back through and look and see what came through the first quadrupole in order to figure out what that was. So I'm going to be using these type of modes a little bit as we go further into the story. Let's first talk a little bit more about flavonoids. These are ubiquitous plant secondary metabolites that exist in virtually any natural product. They exist in a very wide range of structures that are all built from this kind of central A-B ring structure, which can be combined in different ways. And I think you probably have heard of some of these flavonoids before. A lot of these are very desirable nutrient products for various reasons. We're going to focus in on the anthocyanidin, that one up in the middle right top, as a particular flavonoid that confers color to plants, but it has other benefits as well. The anthocyanin refers to the compound where you have an anthocyanidin, the parent flavonoid structure, and then it has appended some different groups there on the bottom, in this case indicated by that glycoside and acyl group. So these really are what provide the reds, the blues, purples, some yellows into a lot of different plants. They should not be confused. Some plants confer plant pigments mainly through things like carotenoids, which are present in carrots. You can get some similar coloring from these different classes, but carotenoids and anthocyanins are very different in their chemical properties. These anthocyanins are well known to have antioxidant capabilities, and in some cases have been reported to have really significant benefit in treatment of different diseases. Now, kind of looking closely to better understand the diversity and complexity of the system, you see here that just general structure on the left, and on the right, you see parent anthocyanidin, so that top part of the left structure. These are different pieces that can form different chemical species that we might see. And if you look at the B ring, the top right ring of each one of these structures, you'll see that the substituents vary. You see, delphinidin has 3 hydroxyl groups, cyanidin 2, pelargonidin 1. Peonidin has a hydroxyl group and a methoxy group. And so already we can see how the diversity of this chemical class can vary just based on the substitution of that ring. And then we'll see later, we can append a very wide variety of different glycosidic groups onto these bottom hydroxyls to create different anthocyanins. The anthocyanins, as I said, are pigments. That pigmentation is dependent on the chemical structure of the anthocyanin that's present, or anthocyanins. It's generally a mixture of them. These are known to even chelate with metals and things. So there's a lot of things in the solution that can modulate the colors that they confer. In this case, on the right, we're just showing how the pH of the medium can affect the color. The anthocyanins are generally stable at lower pHs, below 6, and above there you can have some irreversible changes into what's shown at the bottom there in the bottom right, this chalcone group. So, but even in the ranges of low to medium pH, we can see significant color changes for different anthocyanins if we were to expose them to different environments. And they are present in different environments inside the plant. So this just gives you kind of feeling for how these molecules can change about and change their color. Now, I've had a lot of interest in anthocyanins for a long time. We've done quite a bit of research in the past. Just to give you kind of feeling, blueberries are really highly rich in anthocyanins. We can take blueberries, lyophilize them, which means basically freeze dry them, extract them with some acidic, alcoholic mixture, and then we can take these to our LC or LCMS for analysis. And in this case, not the best separation, but I think you can appreciate the complexity of a mixture here, where we have 25 or so different anthocyanins that are present in this blueberry, and those are listed with their structures on the right. You'll see again these things are depicted as having not the structures, but the names on the right depicted as having the anthocyanin, and then where the appended glycosidic bond is. That can be at these 3 or 5 position. Again, I'll show you a little bit more of that a little bit later. We worked with a company out of New York on this study that makes freeze-dried berries, and we said, okay, well, why don't you send us some of your freeze-dried berries? I mean, we could probably show that the anthocyanins are massively concentrated as you pull out these the water from these blueberries, and it could be a good marketing for you. But when we did the analysis, what we found was that the fresh blueberries, whether they were cultivated wild, had much higher anthocyanin content than the ones that they had dried and or sweetened. So yeah, this was kind of adverse to what they wanted to see, and ultimately they asked that we didn't include their name in the publication, and I understand, but we still appreciate that they played ball with us, and it was interesting to see. So if you're looking at whether to eat your fresh blueberries or your dried and sweetened blueberries, from a nutritional standpoint of the anthocyanins and the antioxidants, go with the fresh stuff. Okay, moving on. I told you, we're using this tandem mass spectrometry, and we're interested in being able to find anthocyanins in these cannabis plants. And so we're going to make use of the well-characterized fragmentation that can occur in tandem mass spectrometry when we use those different fragmentation modes. And this just shows you how different ways that a particular anthocyanin can fragment. So on the left, the easiest things to knock off are these different glycosidic groups, these sugars that have been appended at the 3 and 5 position. And those fall off pretty easily, sometimes even just during electrospray ionization. That will leave you with the residual anthocyanidin, which can further undergo different fragmentation based on, let's say, loss of small molecules or cross-ring cleavages, often referred to as a retro-Diels alder. And this is just a formalism that allows you to name different fragments for these things based on which fragment contains the charge after the fragmentation is complete. So no need to go into great detail here, but just to make it look quite complex. Here for cyanogen are showing some of the fragments that you can generate on the left based on this retro-Diels alder cross-ring cleavage mechanism, or on the right based on the small molecule loss. And I think that the key take home here is that we know the mass to charge ratio, those numbers associated with each one of those fragments. And so we can go looking for those in our sample and have them be somewhat diagnostic of this particular molecule if it's present. And obviously, if we see more than one fragment, that's even better evidence. Just again to show you, if we were to do a product ion scan for these molecules, where we just take those parent anthocyanidins on the top, cyanidin, and the bottom pelargonidin, and you apply these to fragment them and just see what kind of fragments are formed, what you'll see is a whole bunch of signals throughout the mass spectrum that conform to those different fragmentation mechanisms that I saw. And so it's a very rich pattern of information. Again, you can think of this as a fingerprint that we could use to identify those molecules in the plant if they are present. Okay, so let's get into the hemp here. We had worked together with Z Therapeutics. These is a group out of Oregon, Eugene, Oregon, and they provided us with a number of low THC hemp samples. Very, pretty looking stuff, at least, well, I guess when they harvested it. I didn't take these pictures. We get sent them after they've been dried and cured, and they don't look quite as impressive. But from the start, yeah, we can see that there's some significant variation in the colors in these different hemps. We'll take these samples and we subject them to a cryomill, which allows you you can basically put them in this chamber. It has a big ball bearing there in the middle, and then we cool this whole thing down with liquid nitrogen and just shake it up and basically pulverize this sample down into a very fine powder. So you get very high surface area for extraction. So these cryomills are very nice and versatile for preparing samples for extraction because they can make nice such small particle sizes. We take those and then take them and extract them. We're just using a mixture of methanol and water with a little trifluoroacetic acid. We did find that because you co-extract some different waxes, that those could have some deleterious effect on the separation downstream. So what we ended up doing is taking those extracts and defatting them a little bit by extracting that with hexane. So we just, pulling the fats off into the hexane and leaving behind these water-soluble anthocyanins. And then you can see here these extracts and just get a little initial first appreciation for at least here Linny, sherbet, and cattail grape, the different color that you can get from that extract. So we know that there's anthocyanins in there. We just have to go find them. So an example of our strategy would be to go looking for cyanogen. And so we have maybe this parent, cyanin 3 glucoside. And we know if it loses this glycosidic bond in the middle, that we can expect a massive charge of 449. And then if we go further, we can expect some other fragments, these cross-link ring cleavages and whatnot, of some other massive charges that we can look for. So we set our precursor ion scan. So you can see here on the top left the schematic where we've got our first quadrupole scanning from 250 to 850 massive charge. And we're just interested in a signal if something comes through and gives us a signal for 287 massive charge, which is the parent ion for cyanogen. And when we do that, we get this signal, this chromatogram on the bottom, and there's this big hump signal around 12.5 minutes. And then when we go back and look at the first quadrupole to see what makes this, we get the mass spectrum up at the top right. And what we see there is a dominant signal at 611 mass to charge, and this indeed corresponds to a cyanogen 3,5 diglucoside. So we have glycosides appended to both the 3 and the 5 position on this particular anthocyanin, and that appears to be quite a dominant anthocyanin present in this sample. Now, the precursor ion scan is nice for looking for things, but as I said, because we're operating part of this instrument in a scan mode, it's not as sensitive. But we can go now to try to do things to make it more sensitive. First, let's confirm that we're really seeing this molecule and we can generate fragments from it that would be diagnostic what we'd expect. So we can go and do now a product ion scan where we tell the first quadrupole only send through when you see ions of 611 mass to charge. That's the cyanogen 3,5 diglucoside. And now let's use the fragmentation to break this thing apart and see what it forms. So we see a signal whenever we get this thing coming through. Again, the method's changed a little bit, so the time is a little bit different here. But when we see the 611 come through, we can fragment it, and then we can see that we generate a series of diagnostic fragment ions that are consistent with that cyanogen 3,5 diglucoside, and then can say, okay, indeed, that this is quite a large species in the anthocyanin. Now, ultimately, we did this, I'm not going to go ad nauseum, but we did this for a whole bunch of different anthocyanins. And these, as I told you, there's different, anthocyanin parents, and there's different types of appending groups. And what we did is built out a whole series of multiple reaction monitoring schemes for each series of different for each anthocyanin. And so now you see the chromatogram in the middle. This is from an extract of the cattail grape. Each one of those different colors is a different anthocyanin that we've basically created a precursor to product ion transition. Those two filled circles, we're just having our first and our third quadrupole look for specific ions that are consistent with those different anthocyanins. And I think you can see off the bat, there's a lot of anthocyanins in there. And in fact, what we can do is look at that a little bit more holistically and show that we have, pretty large component of, let's say, 3 major anthocyanins, and then, let's say, a lesser amount of some of the other ones. If we do the same thing now, let's say, compare it to now black licorice. Black licorice appears, it's got this really deep kind of purplish, darkish color, really seems to be rich in a lot of different anthocyanins. I mean, even now approaching some of the levels of things like blueberry, which, okay, also has a very deep, rich purple color. So these deep colors tend to mean a more richness of the anthocyanin. We can also project this as a pie chart as before, and you can see maybe a little bit more interesting distribution of some different anthocyanins than we had with the cattail grape, and some different distributions. So we're just starting to now learn kind of what's in these things, and hopefully we'll be able to parse that out further. You'll notice that there are a number of things that are, say, the same thing, right? On the top right, you see delphinidin 611, delphinidin 465, delphinidin 435. I just want to show you very quickly what's happening there, right? So we have this parent delphinidin, which is defined based on having those hydroxyl groups in those positions, 303. And then if we append one glucoside at the 3 position, this gives us mass to charge of 465. So that's our delphinidin 465. Or if we append a rabinoside, so it's a little bit different than the glycoside, which gives us a different mass to charge of 435. So completely different species, but just because it has this different side chain. And there's other ones. So here's a rutinoside, a little bit longer appended chain. And then, as I've shown already, we can have these diglucosides where you have substitution of the 3 and 5 position. And so any of these single substitutions could also occur at the 5 position, although the 3 position is more common. Or you can also have multiple linkages of these glycosides at both of the points. And so this really increases the complexity of the system, as you can imagine. Last one here before I show you kind of a more grand comparison, but Lenny Sherbert, gives us, nice, really red color. And so interestingly enough, the anthocyanin profile is not so complex. This is cyanogen primarily, which, okay, cyan is named for that red color. But just to show you kind of the comparison, at least for these 5 different hemp varietals, and you can see the colors of their extracts, and in each one of these signals, again, is a specific anthocyanin. And so there's some significant overlap and some significant differences, but again, I think this is really the first time anybody's really looked at trying to characterize these colors chemically by profiling the anthocyanins like this. So before I leave this completely, I just want to alert you some upcoming work that's going to be reported out here very soon. We've been working with Zed Therapeutics as well as with Curtis Mathis Grow Lights and Nanova Environmental to conduct a study where we looked at the effect of different lighting on the production of bioactive compounds by 4 different hemp varietals. And so just briefly, what we have here is 4 tent setup. Tent 1 uses a high power white light LED 1212. Tent 2 white light, just with the lower power. Temp 3 and Temp 4 modulate in different amounts of a particular red LED. And the idea here is to perform some phytochrome manipulation and understand how inducing or changing this red light or introducing this red light might affect the expression of different bioactive molecules. So the Nanova Environmental is very interesting. It's a portable GC instrument, and we just basically hook this thing up to the tents, and over time, we're collecting data for a whole series of terpenes. And this is hard to see, but just suffice it to say, tent 1, 2, 3, 4 from the left, and these different colors are showing the expression of different terpenes over time. These are terpene expression. And we do see some significant variation in the terpene expression. Now, these terpenes would be a mass expression from the 4 different genetics that are in the tent. And later at the end, we're doing a comprehensive terpene analysis of the individual plants. But so you see some variation in terpene profiles from the different lighting. There's also some significant variation, although not as drastic, on some of the cannabinoid content and its production. But what we see are, I mean, really just amazingly different looking plants that we can generate from some of these things put under different lighting conditions. We're still working through the anthocyanin data, but just to show you, here's 3 different types of hemp: B73, B120, 246, and these are been grown under the different lighting conditions. And you can see now these are cyanogen rutinoside and pianogen rutinoside, so 2 of the higher abundance anthocyanins, and we can see how they vary with the different lighting conditions in the tents. And we're working to parse this out a little bit further, looking at the full anthocyanin profile, and we'll be submitting a publication on this soon. So as I mentioned in one of the previous slides, or at least it was said out there, you can actually follow this research on LinkedIn by following my good friend and collaborator, Dr. Zach Hildenbrand. So just, this is just the beginning of looking at the hemp color palette. There's obviously, like to apply this across a wider range of genetics. Also would like to be able to widen the net to look at other beneficial flavonoids. The lighting study, I think, is a really interesting add-on and should provide another tool for people cultivating hemp to modulate the bioactive components. We have started looking at wider range of things in these hemp, but we got to start someplace, and the anthocyanins certainly are an interesting area to look at, because I think everybody can appreciate the varied interesting colors that these plants can take. So I would just like to end by thanking collaborators, Dr. Zach Hildenbrand, Adam Jacques, and Christian West of Zed Therapeutics. We've also gotten a lot of support from Shimadzu Scientific Instruments and ResTech Corporation. And I want to thank Lab Roots Conference for the opportunity to present this work. Thanks very much for your attention.